today's episode. So, what are the learning outcomes of today's lesson? So, at the end of this lesson, we will be knowing that what is a dihybrid cross and what are the interpretation of Mendel's observation. So, Mendel's observation not in this dihybrid cross but also in the monohybrid cross, what he observed, we will be reading or learning that thing also in detail in this episode. So let us start with dihybrid cross. Now before that, I would like to ask you a few questions that whether you remember monohybrid cross or not. If you have not gone through that monohybrid cross lessons, please go through it. I am putting my link of monohybrid cross lessons up, and please don't forget to go through those videos. If you are not reading the monohybrid cross first, you will never understand this dihybrid cross. So first go through it. So if you are very much clear with monohybrid cross, then only you can proceed with my lessons. So let us move on to today's dihybrid cross episode. So what exactly is this dihybrid cross? If you remember the monohybrid cross, you will remember that there was only one contrasting character. One plant we have taken tall, another was short. But here we are taking two varieties of pea plants having two contrasting characters. There we took only tall and short plant. We took only purple flower and white flower. That too separately. So it was only one contrasting characters which we have taken in monohybrid cross. Now, if you are going to dihybrid cross, what is it? It is the one in which two varieties of pea plants are taken and which are having two contrasting characters, which are crossed to study the inheritance of two pairs of traits simultaneously. So, what we are doing? We are crossing the two contrasting characters to see what is happening in the F1 generation and what is happening again in the F2 generation. So in monohybrid cross, what we have seen, so just a quick recall of that, we have seen that in F1 generation all the plants were tall, but in F2 generation few were tall and others were short. So the recessive character or the character which was suppressed in the F1 generation came out in the F2 generation or the second filial generation. So let us see what is going to happen in this dihybrid cross. So this we don't know. We have taken only two contrasting characters and they are crossed. And now we will study what is happening in these two pairs of traits after being crossed. So let us start to see what is happening here. Here we will be discussing about the dihybrid cross. Now first let us see what are the characters we have taken, contrasting characters. We have taken yellow and round, and this is green and wrinkled. So these are the symbolic representation of yellow and round, that is capital Y and capital R, and this is green and wrinkled, that is small y and small r. Now these two have been crossed, and in F1 generation what we see that the offspring what we are getting is having yellow and round seeds. So after first the F1 generation or the first filial generation, we are having yellow and round seed. So here we can see the small y and the small r characters are there, but they are in recessive form. That is, they are being suppressed by the capital Y and the capital R. So which we know are used to represent the dominant characters. So I think you still now remember the first episode of Heredity where we have discussed about all the dominant allele, recessive allele, homozygous, heterozygous and all. So if you don't remember, if you want to recall it again, please visit to that video. I am putting the link here on the top. And don't forget to again I'm telling you to recall those things so that it should be very clear for you to read this dihybrid and monohybrid cross. And you should never memorize this. This is not a thing to memorize. You should understand. So if you understand it, then only you can continue with different sums, or we can say you can solve different complex problems of genetics. So now what is this? We should come to this one. So this is something which is looking, we can say it is a square shaped thing. It is a square shaped, what is it called? So it is called Punnett square. So why is it used? So if we are placing few gametes here, and if we are placing the other gametes here, what is happening? So Punnett square, what is it? It is a diagrammatic representation. We are representing all the offsprings, all the genotypic combination of the gametes from both the male and the female parents. So these are gametes, suppose from the male parent, and these are from the female one. So when we are doing the crossing over, this is the offspring. So we can get the genetic combination of the offspring in this Punnett square. So this is how we are diagrammatically representing. So this is a simple mathematical calculation, and we can do many problems of heredity with the help of this Punnett square. So now we will see this in this Punnett square. So what is happening here? So we have taken YR here. So this is the male part and this is the female gametes. So if this capital Y and R is being crossed with this YR, so we will go in this way. So this is all the columns and this is all the rows. So YR into this, crossing over into this YR is YYRR. So again this capital Y and R is being crossed with small y, capital R. So what we are getting? Capital Y, small y, and capital R, small r. So this thing goes on. Again this one is being crossed with this one. So capital Y, capital R and small r. So this thing continues. Now the same thing will happen when we come to the next row. So here we can see that the female gamete is small y, capital R. So this will again be crossed with the same male gametes. So capital YR, capital Y, small r and small y, small r. So these are the genetic combinations of the offsprings. Same thing happens in the next row and the last row. So this is all what is happening in this Punnett square, which is a simple diagrammatic representation. And then we can see that what is happening here. So we will get the possible combination of genotypes and the resulting phenotypes can be concluded. So here what is the phenotype and what is the genotype? So that we will discuss. So first let us see what is happening here. Now for example, we have taken two plants. In the F1 generation we have seen that yellow color was dominant over green, which is a recessive character, and round shape was dominant over the wrinkled one. Now again in F2 generation, Mendel self-pollinated these hybrids obtained in the F1 generation. So when the F1 generation seeds were crossbred, in F2 generation progeny they showed 4 different kinds of phenotypes of the seeds. So what it was showing? Round seed with yellow color. So this is yellow color and round seed. They have shown wrinkle seed with green color. So this is wrinkle seed with green color. Okay, is it clear? So wrinkle seed with green color. The color is green and the seed is wrinkled. Now, but the two new combinations appeared were round seeds with green color, and we have seen that wrinkle seeds with yellow color. We have seen round seeds with green color and we have seen wrinkle seeds with yellow color. So these two combination was new. This was not known that it will come out in the F2 generation or the second generation. So first one, we observed that round seeds with yellow color. We observed that we have got that both the parents, though they were having round and yellow color, they were wrinkled and they were green in color, but we got new two combinations. So what were there? That is round seeds with green color, round seeds with green color, and we got that wrinkle seeds with yellow color. So now there were yellow round, yellow wrinkled. So yellow round and yellow wrinkled. So all the characters are mixing here. You can see green round and green wrinkled. So yellow round, yellow wrinkled. Green round and green wrinkled. So the ratio of dihybrid cross was 9:3:3:1. So the dihybrid cross ratio was 9:3:3:1. So please make it clear until we are moving to the next slide. Please make it clear what is happening in this Punnett square, how we are crossing this gametes with this gametes. So how this offspring gametic combination is there, how we are getting the phenotype as well as the genotype. So please go through your textbook. Whichever textbook you are following will have this in details. After reading in my video, please go through the textbook also, so that you get a clear picture of what I have taught. So now let us move on to the next one. Here comes the interpretation of Mendel's observation. So after two crosses, monohybrid and dihybrid cross, what did he interpret? So let us see here in this Punnett square. What did he interpret? So when we have taken monohybrid cross, when cross is made in between contrasting pair of one trait, so this is only tall plant and this is only dwarf plant or short plant. So what is happening after crossing? So we have seen that when we are crossing the male gametes with the female ones, you can put it vice versa also. Here you can put the female gametes and here you can put the male one. So it doesn't cause any variation in your genetic combination here. It will be the same because you will be crossing the rows and columns. So now when we are crossing the rows with this column, so we are getting TT. Again this one with this one we are getting TT. This one with this one TT. And this one with this one capital T small t. So what is happening after crossing in the first filial generation? We have seen only one is dominant. So all the four plants were tall. So the small t or the recessive character appeared, but it didn't show up, or none of the plants of this generation were tall. So all were dwarf plants. So which was the trait which was not present in the offspring of a particular cross in F1 generation again reappears in the F2 generation. So this is what we are discussing about the F1 generation. Now let us move on to the next part, which was the F2 generation, where this recessive characters reappears. Though it was not there in the first filial generation or the F1 generation, it started reappearing in the F2 generation. So let us move on to that one. Now here after crossing this, we can see that what we got from the F1 generation, we are crossing those gametes. So this one and this one in Punnett square, we are seeing the result is capital T, capital T, capital T small t, capital T small t and small t small t. So what we will say? So interpretation of monohybrid cross was genotypically their ratio was 1:2:1. But if we tell the phenotype, that will be, we will see that 3 plants were tall and 1 plant was dwarf. So the ratio is 3:1. So 3 plants were tall and 1 plant is dwarf plant. So in F2 generation, this dwarf plant appeared as a new character which was recessive in the F1 generation. So this was particularly, it is reappearing in the F2 generation. This is the recalling of monohybrid cross again. And I am telling you again and again, please make it very clear so that with the help of this Punnett square, you can solve any complex problem of genetics. So it is very easy. Just you need to cross between rows and columns, and you need to place the gametes in these two boxes separately. So these are all alleles, right? This T, this capital T is the dominant allele, and this small t is the recessive allele. All has been described in that heredity introduction part. So please again and again I am telling you, requesting you, go through it. And if you have not yet subscribed to my channel, please do subscribe to my channel, and don't forget to like and share my videos. Now let us move on to the interpretation of the dihybrid cross. Now what did he interpret in this dihybrid cross? In this dihybrid cross, he said that when a combination of contrasting pairs of two traits were taken together, what happened? Only one variety of each trait appeared in F1 generation. So that we have seen in the Punnett square earlier. Again, we will do a revision of that Punnett square at the end. So what happens? The other variety of each trait, it reappears in the F2 generation. So as almost it is similar to that of monohybrid cross, there it was only one contrasting character. Here a pair of contrasting two traits were taken. So it reappears in the F2 generation. And the presence of two new combinations of two contrasting pairs of traits in the F2 generation also occurs. So this is a new thing which is happening here. So this we have seen there, what is happening? We have seen there that new thing, what is happening? We have seen a new combination. What came out is round seeds green in color and wrinkle seeds yellow in color. So this is a new combination of two pairs of contrasting characters which came out in this F2 generation. So let us quickly look at the Punnett square again. Now here just a quick revision. We have learnt it earlier. So yellow and round being crossed with green and wrinkle seed. So what is happening in the F1 generation? So yellow and round seeds plants are formed. So which are having green and wrinkle character as a recessive. So after this crossing in the F2 generation, after self fertilization, in the F2 generation, what we are seeing, the ratio is 9:3:3:1. So we are seeing that round seeds with yellow color round seeds and wrinkled yellow color seeds are there. So wrinkled yellow color seeds are there. But it should have been that round yellow color seeds and wrinkled seeds with green color. But we have seen two new combination, that is round seeds with green color as well as wrinkle seeds with yellow color. This new combinations has appeared. So how come the ratio is 9:3:3:1? So here you can count 1, 2, 3, 4, 5, 6, 7, 8, 9. So these are all yellow seeds having round shape. So 9. So 3 is yellow and wrinkled. So 9:3. Now another 3 is round but they are green in color. Okay. So 9:3. This 3 is to 1, 2, 3. This 3 is to 1 is, that is green and wrinkled. So the ratio in this Punnett square is 9:3:3:1. So we can see here the phenotypic combination as well as the genotypic combinations. So please try to understand this Punnett square so that in near future you can do all the complex problems related to genetics. So this is all for now. This is all for today's lesson, and we will be meeting soon with all the 3 laws of inheritance by Mendel. So let us complete today's episode with this dihybrid cross Punnett square. So again I am telling you that please do go through your lessons and don't forget to subscribe to my channel. Thank you.